also from Oxford by Vicente Grau about image-based cardiac modeling.
Thank you very much. And again, thanks to the organizers for putting this together and for inviting me to give this talk. I'm just going to give this, first thing I would like to say is I'm just going to talk about a larger project, and I'm just a member of a big group, and there is a lot of this that comes from Peter Cole's lab, several people at Computing Laboratory, and particularly Martin Bishop and Gernot Planck for what I'm going to talk about today, and lots of the imaging from Jürgen Schneider's lab, everybody here in Oxford, and we also have Natalia has also been involved in this project from the early stages, and there are some other collaborators that I am probably forgetting. Now, many of the whole heart models that we are seeing and that have been published use a simplified, highly simplified geometry, and this is okay for. I mean, it has produced very, very good results up to now. However, we all know this is not the real situation. This is just one of the MRI slices that we are working on. And this is not just a matter of putting these into the models. And Natalia was commenting on that before too. If we are going to go towards patient-specific or personalized modeling, then we are going to need, we are going to use clinical images. And clinical images actually resemble this much more than this, at least with the current technology. So we are sort of stuck with this for now, but we don't really understand that well what is the effect that simplifying this very complicated geometry to something like this, what is the effect that this is producing. And this is something that we are trying to understand by using different types of images and building models. Well, this is the pipeline approximately. It just changes with time. But this is more or less the pipeline that we are using right now in terms of image acquisition. So we have different types of images, including MRI and histology, and some of them are in vivo and some of them are ex vivo. So starting from the left, we are acquiring scene sequences, and that would be what simulates approximately the clinical scenario. So we have this beating heart MRI. Here we have short axis slice, about 1/10 of a millimeter is what we get for a rat. This is a rat's heart in this case. This is a transverse slice. So this would be a long axis, and I didn't interpolate it or anything. I could have made it nicer, but I just wanted to represent here what is actually the amount of information that we get, because we have about an order of magnitude worse resolution between slices than what we have within a slice. But this is the only image, the only scan where we have the beating heart in vivo, and then we are going ex vivo. We are acquiring diffusion tensor MRI images. I should mention that all of this comes from Peter Cole's and Jürgen Schneider's labs. So we are acquiring the diffusion tensor MRI. Here we have about 1/10 of a millimeter isotropic, and we are getting the myocyte orientation. Now we are ex vivo. Here we are acquiring high resolution ex vivo MRI images, and these are around 20 micron isotropic resolution. And here we can see all the small vessels. We can see endocardial tuberculations. We can see very well what is the aspect of the endocardial wall, and so on. And if I can move to the next one. Now this is the best in terms of resolution and tissue identification. This is the same heart, but now these are histological slices acquired at, in this case, this one is a rabbit heart, I have to say. This is 2.5 micron in slice and about 10 micron between slices. And it's not just that we have a much better resolution here, but we also have lots of things that we don't see at all in the MRI. Now these images, because they are slices, they are misaligned, and they have suffered individual deformation in the process. But we can see things like, if we focus on a small area. We can see different types of cells, and we can see very much paracellular resolution. So the idea is, how can we combine all the information from this? If we look at this pipeline, at these different types of images, we are going from left to right. We are increasing in the level of detail, and we are increasing in the information that we get from the images generally. But we are increasing also the geometrical distortion. So at this side, where we have pretty poor resolution, it's the only place where we really have the geometry of the original heart, and the only place where we have information about the motion, which is what we can use to validate the models. And on this side, we have the worst amount of distortion, but we have the best resolution, and this is the best source of information in a way. So to go back to my first slides, then what, as I told you, what we want to do is to quantify what, by simplifying these models, what we are doing, what source of error we are introducing. And I'll be focusing now on this part, on the MRI images. So we take the original MRI images. These are ex vivo, as I said. This is about 25 micron isotropic resolution. We are doing a segmentation. I will spare the details, but it's far from straightforward. And the thing with segmentation here is that very small errors in segmentation can change totally the connectivity of small areas. So we have to be aware of that. Then we are segmenting different structures, papillary muscles, valves, and so on. We are generating meshes, and this is mostly the work of Martin Bishop with a lot of input from Gernot Planck. Then we are introducing fiber orientation, and in this case, we are doing it by using a mathematical model validated by DTI images. And using, we define the fiber orientation in the papillary muscles as parallel to the main axis. And this is the important bit. So we get this detailed model using all the information that we have in the images. We have the valves, we have all the big and small vessels, we have endocardial trabeculations, and so on. But we are also building a simplified model from exactly the same images by changing the parameters of the segmentation and doing a couple of post-processing stages. So we have a very detailed model, and we have a very simplified model, which is sort of what we could get, about what we could get from clinical images. And we have done some simulations, and this is quite preliminary, so I wouldn't want to reach too many conclusions from this, but this is exactly the same protocol. This is a single apical stimulus, and if it works, yeah, it does. So we see it's approximately the same. On the top, we have the detailed model. On the bottom, we have the simplified model. You can see how we have, in the area close to the septum, we have an early activation. This causes a change in the speed altogether, but it is not, the local effect is much more pronounced. And this is, we think, is caused by the trabeculations, and the cardiac trabeculations are acting as short circuits. And locally, it is really dramatic, the difference that we see. Now, this is, of course, not unexpected. This is a little bit more intriguing. We are applying a simulation of a re-entry, both to the detailed and to the simplified model, by an initial stimulus at the apex and then an anterior-posterior shock. And we also see very clear differences between the detailed and the simplified model. And in this case, it looks like the detailed, we get a very clear, well-organized single wave. And the simplified model, the wave fractionates very early on in the simulation. And as I said, I wouldn't want to discuss what might be the reasons for this yet, because I think it needs more. It's basically opening a lot of possibilities for studies here. We really need to look into this more, but it looks like there are very significant differences using exactly the same protocols and with the same geometry. So this is just using these images here, but we are still missing a lot of information that we have in the histology slices. And the problem with that is that the histology slices, because they are individual slices, they are totally misaligned, and we cannot use them to build models right now. So we have been working on how to introduce the information from histology in these models, and we should be able to use to introduce different types of cells and things like that. This is the process, and there is a different embedding for getting the MRI images and for getting the histological image stack. So what we have done, we have developed this co-registration technique, in which we have here an iterative procedure in which we basically try iteration by iteration. We try to align in 3 dimensions the MRI volume and the histology volume until we can find slices in the MRI volume that correspond optimally to the histological slices themselves. And once we have done that, we align the slices one-to-one. We do a one-to-one, 2-dimensional, non-rigid alignment. This is working. This is, again, the MRI image. And here we are comparing it with what, so this is the original histological slide. We are comparing it with what we have found to be the best MRI slice, right? It's not necessarily one of the original slices. It can be a diagonal one. And we see an approximate correspondence, a pretty good approximate correspondence. And we also see small parts where, as we would expect, there are small deformations. And we are still working on ways to correct that. These are other examples in which we see approximately the same things. And in some cases, we actually have important differences, and this is in part due to our registration, but in part it is just due to the fact that the data sets are different. But hopefully we are close to introducing this information into the models that I showed before. So the conclusions, this is more probably more answers than questions here. The other way around, more questions than answers. So current imaging techniques offer the possibility of producing detailed, accurate models, merging multiple modalities and acquisition types. And here I'm talking about histology and MRI, but of course there are other clinical and research imaging modalities. Now the image analysis itself, the image processing part, remains an important challenge. I didn't talk about that too much, but it is far from straightforward to generate the models from the images. We see all sorts of artifacts. Now, the extent to which small structures affect different activation patterns, we seem to see in our result that this actually produces a pretty big effect. Now, of course, the question is, in which applications do we need to take that into account? And for which applications is it just okay to use a very simplified model? But if there are applications in which we really need to put all this information in the model, then we need to find a way to include this in personalized models, because we are not going to get this information about things like endocardial tuberculation from personalized images in the clinics. Thanks.
This is very nice work. I like it. I was thinking that the histological section is probably a curved surface through your MRI data, isn't it?
Yeah.
Or if you tried to find that surface.
That's we are aware of that, and we are assuming that they are not, and that's because the problem is still posed from the beginning, and it becomes terribly under constraint if we just if we allow from the beginning any sort of 3-dimensional deformation, then we can just it just won't converge to anything sensible. So yeah, we have to introduce we have to introduce these sort of constraints, and we are aware that they are not exact.
Okay, so I would like to thank everybody because we're a little bit behind time, and maybe we will shift discussion in